Okay, I am going to start recording. Okay, I'm going to go ahead and get started so that we have more time for discussion. First, my terrible joke of the day. Sorry, I'm accepting all of the AI note takers. Terrible joke of the day. Who is the smartest pig? Einstein.
That's a good one.
It really is.
Ein Schwein.
Then today is a very special day because not only is Kai presenting, but we've also invited the group from Cure GABA A. So I am gonna go over our typical notes, but rather than go through an entire round of introductions, we were hoping today could be more interactive and that people could introduce themselves as they ask questions. Or if you have a question, put it in the chat with a short introduction so I can keep my eye on it too. For those of you that don't know me, my name is Amber Freed. I founded SLC 6A1 Connect. We're a patient advocacy group dedicated to improving the lives and children of those impacted by SLC. And we raise money. We advance scientific research. We spend all of our time organizing. We are going to have our big symposium later this year, the day before the official start of AES. I will put a link to register in the chat. I will also put a link that if you'd like to be invited or like to be included on our monthly mailing list, we would love it. We don't stalk or spam or anything. And you can also add your name and information to our Google Doc if you would like other people to be able to get in touch with you just from this lab meeting. There is new research this month. It's not peer reviewed yet, but it is on phenylbutyrate and the effect on fruit flies. I will send out the link to that paper along with the wrap up from this call. There is a grant actually, and it is from the Sergey Brin Foundation. These are $15 million grants to hospital networks to become essentially clinical. Or Dennis, am I misrepresenting when I say to become clinical trial ready? To identify autism patients, enroll in bulk with the end goal of becoming clinical trial ready.
Yeah,
RFAs, oh, good.
yeah, so it sounds good. So become a clinical trial site, enroll 250 people over 5 years and be able to deep phenotype them.
Yeah. It's so exciting though to see some of these private organizations like stepping up in a really big way to fill some of the holes we're seeing in government spending. Next month, we will welcome Chris McGraw. Many of you probably remember him being at Harvard. He has moved to Northwestern, and he is going to be giving an update of prior a prior presentation on GABA fluorescence as a functional assay for GAT1 variants. And lastly, one side note that I just wanted to add, at the prior lab meeting, I think it was prior, Lainey Mosley talked about her documentary of rare disease families and their journeys and being thrust into the situation where you're a caregiver and scientists and everything all at once. Lainey actually previewed, or not previewed, premiered her movie at Harvard in thanks to Henry Lee, our big supporter on this call. Thank you so much, Henry. And it was just an incredible success. Like, I can't say enough good things. There was a panel afterward and a reception. And I think the more and more of events we have like this that are just educational and human, the better. I keep saying this, and I'm going to keep saying this, but the rare disease movement is a lot like HIV and AIDS was in the 80s and 90s, and we're finally at a place where we can do something about this. And it's going to take a whole lot of people understanding the problem and understanding the solution as it is now. Last week, SLC was able to bring in some moms for a panel at UT Southwestern, and it was just a great day. Matt Rue was there on the call. Ray from UT Southwestern on the call was there. And it it helps scientists see why they're working so hard, and patients appreciate the opportunity to tell their story. If your institution is interested in organizing an event like this, I think it is just so crucial for everybody. A rising tide lifts all ships. And it's also been very good for fundraising because universities, if they're going to take the time to host some type of an event, they typically invite people that are donors to the institution to see where their money goes. And I'm all for helping all of us. So if that makes sense, please reach out to me. And with that, I will hand things over to Kai, unless anybody has an announcement they would like to make. Okay, so I am going to stop sharing. Kai,
Okay.
if you would like to begin sharing
Yes, sure.
and introduce yourself, and I will put some links that I went over quickly into the chat. And like I said, if you have a question, feel free to put it in the chat or raise your hand and introduce yourself, and we will call on you. Take it away, Kai.
Okay. Right. Let me see. Yeah. I should get this. Yeah. Okay. From here. So okay. I'm a physiologist and neurobiologist, and my most of my actually I could start and tell something about this. Can you see my slide? Can you see this? Yeah. Okay. Yeah.
Yeah,
So
all good.
right, yeah. So I've been working on quite a few things like physiology and neuroscience and so on. But I mean, during the recent days, well, years, developmental aspects have been in my focus, and lots of like disease-related research as well. So like birth asphyxia, febrile seizures, mechanisms of seizures, and manifestation of epilepsy. And then more recently with Michel Monge, some pediatric neuro-oncology. So there are like really interesting like binding themes through these different branches and so on. And I could of course talk about them, but I mean I'm like slightly pressed in time, so I go directly into the subject matter. So I see quite a lot of real specialists here in the audience. I try to put the sort of the level of the talk like on a well such that even a layperson could actually follow a little bit like what I'm saying here. So I hope this is not a big disappointment to those who are at the cutting edge level. So anyway, so I like the concept of development of health and disease. I think it's extremely useful. So if you think of plasticity or actually life, so there's developmental plasticity, which is early on fairly wide, and then it gets more constrained during the course of life. Also, like incidence of or chances of risk of serious illness, they actually, well, there's obviously it increases during one's lifetime, and it starts already during the prenatal period. Actually, the prenatal period is extremely important. So we already have one life here before birth, actually. Yeah. So here. So lots of things happen there. And of course, like mutations have a huge effect on developmental before we are born. Then things go fine, so no problem, low risk trajectory here. But then if something goes wrong, like for instance, during the prenatal period or perinatal period, things can go to this sort of a high risk trajectory shown here. So high risk to get like a serious illness at some stage of life. And usually these things are additive, like and multiplicative even. So when things go wrong, they just start to go even worse. So and this has practical consequences. So if we have early interventions during the high plasticity phase, there's much more that we can do. So the chances that we actually get sort of the trajectory of development back to normal is higher to highly functional level. Well, later on we can do something like some interventions, but we don't necessarily get anymore to the optimal sort of functional trajectory. And then, of course, with worse insults and so on later on, the chances are even smaller. So early interventions are, should put this somewhere away. So early interventions are, they are more well effective, of course. They can be curative, which means actually that the only preventive and curative therapies which we really can have in major CNS diseases are those we can implement early on. And then later on, the interventions get symptomatic. So we are actually dealing with symptoms and sort of mitigating symptoms rather than curing disease. So I actually like the slogan, "We cannot wait." This has so many dimensions in rare diseases and, of course, in neurodevelopmental disorders as well. All right, so let me see. Yeah, so a couple of words about development of GABA-A receptor signaling, which has been a key area in my research. So I'm not going into details here, but we know that in the immature neurons, and I guess like 99% of the audience knows that in immature neurons, GABA is not hyperpolarizing. So initially we have the transporter there more or less like heavily expressed, which pumps in chloride into the neurons, and it's driven by sodium. So all the ion transport is actually parasiting on the mother of all ion pumps, the sodium potassium ATPase. Anyway, we have active uptake of chloride into neurons, and as a consequence, if there's any sort of pathway conductance pathway out from the neuron, we get the efflux of chloride, which of course means depolarization of the cell membrane, which sometimes is excitation, not always, but sometimes. Then we have the KCC2 transporter, and which is pumping out chloride using the potassium gradient, sorry, created by the NAK ATPase. And there's chance after this active transport or during it to have a conductance pathway that is open, chloride comes into the cell and produces a hyperpolarizing current. So what we found very soon after the KCC2 functions was that in neurons, if they get damaged, KCC2 is lost very often. And this is actually an adaptive, most likely an adaptive response. But anyway, it leads to depolarizing GABA responses again, sort of in damaged, more mature neurons. it's simply because now we have the opposite. We have NKCC1 upregulation and downregulation of KCC2. So this is part of a much bigger sort of de-differentiation picture, but the electrophysiological facet is seen here as in a very sort of simplistic manner. So neurons cannot divide; they have to do something else to survive trauma and damage. So. Right. So in order to do sort of serious animal research related to, for instance, neurodevelopmental disorders, we have to have a calibration. So from our rodent models. So these are rat pups here. So we take, because of many properties of the brain, of the nerve cells, molecular markers, and so on. We take the P12 postnatal day 12 pup as a sort of equivalent to the term human neonate, and then the postnatal day 0 pup is equivalent to a very early preterm baby. So and things seem to work pretty nice in this calibration. So we see in both in pups, neonatal pups, if you use DCEEG, the very low level high pass frequency. This is actually DCEEG. We see in human preterm babies, we see extremely massive EEG transients. They have up to 1 millivolt or even 2 millivolts, like an amplitude, and you can see that the time basis is really like slow here. So these are extremely big spontaneous activity transients. These are abolished, interestingly, in both rodents, and this kind of activities abolished during the upregulation of KC2, as shown here in this colorimetric in situ graph. So and of course we see also, like, I mean, this is contested by many people. I don't know why, but we see KC protein in term neonates being being like salient, with salient expression in the cortex. And some people claim that KC2 is upregulated by the end, like actually is functional by the end of the second or first postnatal years, which of course can't be true. And okay, so I'll just proceed from here. So GABA has many phases as a signaling molecule, and I'm touching only on one sort of slightly different function here. So we found somewhere in 1998, we found that GABA can be a bona fide excitatory transmitter. So if you just stimulate in a slice preparation, if you stimulate interneurons, like shown it, can you actually? I have this sort of zoom thingy here in my view, but I mean, can you see the slice preparation down there? So anyway, so what we do is we stimulate in the presence of glutamate blockers interneurons close to the pyramidal neuron wave recording. And what we saw was that with a sort of a typical like high frequency burst, like 40 pulses in 40 pulses at 100 hertz frequency, we saw that at P11, at postnatal day P11, there was no after discharge, but at sort of later age points, like already at P12 in this litter, but at P15 and P25, you can see that there's a massive after discharge, which is typical of it's an element of seizure-like activity. And it was interesting because this is fully dependent on the presence of bicarbonate. So bicarbonate acts as a very powerful carrier of current in GABA receptors. People hate the idea that actually a significant part of GABA receptor mediated current is actually carried by bicarbonate, because it makes things complicated. And my sort of I got the hunch that it's getting more and more like common that if people don't like a fact, then it's then not a fact anymore if you don't like it. But sadly enough, it's the GABA-A current is much more like soft, sort of complex than a simple current. So here we see on the right, there's the power of this after discharge. So we had benzoylamide, that's Ba, and ethoxyzolamide, which are membrane impermeant and membrane permeant carbonic anhydrases. So what you can already see from these graphs is that applying a membrane permeant carbonic anhydrase blocker strongly suppresses this kind of a epileptiform after discharge. So that was a finding that led to a lot of other findings. So the scheme is here. So basically, what happens? So we have the basic ion regulators here, but then we have bicarbonate flux coming out of the cell because the pH regulatory mechanisms of a neuron. A neuron doesn't have pH at an equilibrium. It has a more alkaline pH than thermodynamically passive distribution would mean, and therefore you have always a bicarbonate efflux through the GABA-A receptors. And the crucial molecule here is carbonic anhydrase in the cytosol of neurons. So it's here. So the first neuron carbonic anhydrase isoform is CA7, which is neuron specific, which is quite interesting. And then after that, we get CA7 expression. So now if you look at the sort of comparison, interspecies comparison of like a lab rodent and human, how these actually work. So initially, during early development, we have a mainly like NKCC1 dominated. It doesn't have to be really excitatory, but deep rising GABA response during the upregulation of GABA. So this is functional expression in neurons. So we have upregulation of KCC2 and of course efficacy of chloride extrusion. So we get these classical hyperrising GABA currents. But at the time of birth in humans, that's actually quite interesting. So perinatally, this is unpublished, but perinatally, and this happens in rodents at the equivalent age point of P10 to P12. We have in humans, we have expression of this neuron-specific carbonic anhydrase CS7, and I'm sure it does a lot of it has many functions in neural networks in the perinatal and early neonatal brain. I will come to them in a suitable time slot, maybe later on. And then we have later, I mean, in development here, we have expression of the sort of household isoform of carbonic anhydrase CA2. Okay, so so there are at least like 3 different ionic mechanisms which govern GABA transport during development, and of course these change in damaged neurons. Right. What about extrasynaptic GABA? So my focus should be now on tonic GABA because we are talking about the SLC6A1, which codes for GAT1. But the ionic mechanisms I just explained, they of course apply both to phasic and tonic inhibition. I think this is important take-home message. So if there's deep rising, if there's a deep rising phasic GABA response, there's also a deep rising tonic GABA response. So Steve Brinkley and Ishwan Mori were the first ones actually to really identify tonic inhibition, and this is a review they wrote together much later on in 2011, and they had this scheme in this review. So on the left you see phasic inhibition. It's getting like if you block GAT1 here, you get if you have GAT1 function prominently here, you get a prolongation of the iPSC seen here. So these are phasic responses which are prolonged if you have lots of extra synaptic GABA around. And on the right, you have tonic inhibition, which is basically just a baseline current, which of course affects only also baseline conductance. And the exocyanic receptors, they have different composition, but I can't go into detail here. But anyway, we have ways to differentiate experimentally between tonic and phasic inhibition. So okay, so this is what is controlled now here by GAT1. Importantly, by the way, if you think of the time integral of total current of phasic and tonic inhibition, fact is that in fact, tonic inhibition mediates in most neurons, it mediates more current as a time integral than phasic inhibition. So it's a really strong modulator of neuronal excitability. Okay, then closer to our sort of research topic, like actually the SL61A mutants. So there were several papers by Isham Moody and Henry Lester and their colleagues on the GAT1 knockout mice. And these papers are actually worth a second look even today. They were really nicely conducted experiments, highly sort of informative. So the mutations, this crowd here, I mean, those who are working on the GAT1 are mainly they are loss of function mutations. So obviously they are comparable to knockouts. So you can see effects in the heterozygote knockout and in the full knockout, and as pretty much similar results as gained by Istvan and Henry are seen in these mutant animal models. So they have tremor, ataxia, nervousness, and so on. And quite a lot of these phenotypes are based on dysfunctions in the cerebellum. And this is also seems to be clinically the case. So, so too much GABA in the extracellular space modifies circuitries in a radical manner, as shown already in the title of this paper. So there was a recent one, a very beautiful paper from Kathy Kang's lab. They looked at two mutants, heterozygotes, and I would say that quite a lot of phenotype similarities were seen with the knockouts. So this is an interesting point to compare. And the loss of function in these mutants is basically related to mainly to sort of lack of functional trafficking to the membrane, which is a very common sort of loss of function underlying cellular mechanism. So what about the development of GAT1 in the human? So the stippled line there is a rough approximation how GAT is actually upregulated in our brains. So you can see again that it belongs to the same gene family as KCC2. So there's a cluster of genes or clusters of genes which have been studied, for instance, for the first time by Nenad Sestan. He had a breakthrough paper in Science in Nature in 2016, and you can, well, I mean, there are lots of places where you can data mine like expression patterns. Like since this 2016, you have hundreds of databases where you can mine the expression patterns. So this is from like one of these reproductive data banks. Okay, so we have at birth we already have a lot of GAT1, and I wonder what the mutant does at the perinatal and early postnatal period. So I promised to be very fast with my talk, so I'm already coming to the final section. So quite surprisingly, Guillaume Melikishvili, who gave a talk in April, he and his colleagues found that acetazolamide, which by the way is one of my favorite drugs because I mean we used it for seizure suppression in a model of birth asphyxia seizures in a successful manner. I was delighted to see their paper on CHD2, where they also showed that acetazolamide is actually efficient in those patients to suppress seizures. But it seems to be effective also in this SLC 6A1 related epilepsy. And now just remember that I showed a couple of minutes ago that acetazolamide and other carbon anhydrase inhibitors, they remove the excitatory component of or the bicarbonate component of GABA transmission. So that actually might be one of the underlying mechanisms. But really, I mean the sort of the therapeutic effect of acetazolamide is really astonishing here. Those who were present at Gia's talk, they got more of the details, but I just compared these two EEG recordings before ACZ and after, or actually during therapy with ACZ, and you see the massive differences. These are, in fact, flickery vote seizures, which are like very sort of a useful indicators of seizure susceptibility. Okay, what I've been wondering now for quite a lot of time is that how come it is that so many different neurodevelopmental disorders with totally different genes, they actually converge to similar phenotypes. So is that just a sort of, is that our way of classifying disease, or is it something else? What are the underlying like biological factors? I think this is a well-recognized problem in this field of research. One can, of course, speculate a lot, like if you give the brain a hit, it tends to go, well, it tends to have some sort of a compensatory or adaptive set of mechanisms at different levels of organization. And that, of course, is a very sort of broad explanation what happens. But let's look at this here. So Nenad Sestan had I think, a really informative scheme in one of his papers. Sestan has stated some time ago. The genes here and are inserted by myself, but I mean otherwise, like I mean this gene, protein, cell, circuit behavior. That scheme per se is from Nenad's publication. So it is actually quite stunning that all the mutations in all the genes shown in red, the phenotype is mitigated by acetazolamide. So this is a huge string from SLC 6A1 to CACNA1A, GLUT1 deficiency, CHD2, and lots of other ones like which were added more recently. So all of these are mitigated to some extent by acetazolamide. And one wonders what is the reason. So the proximate causes of these mutations are most likely not like sort of rectified by exposing the organism to acetazolamide. But then the convergence, it can be something. It can be, well, I'm just postulating, it could be this like excitatory kappa stuff. It could be overall excitability of neurons. We really don't know. And this is why I'm like, well, we get these CL6A1 mutant mice to my lab in a few weeks. So let's see how we start to dig into them, like looking at of course at GABAergic transmission, but also behavior and so on, and try to find out what these future potential therapy really is about at the level of mechanisms. So I'll just stop here, and I had quite a lot of fun. Been lucky, I have a lot of good collaborators. They are on the right, my long-term collaborators, and then the current project, Guillaume Melekishvili, Pasquale Striano, Olivier Dula, and Otto Goniashvili, and many others have sort of been like doing the clinical work, and Amber has been like well, I mean, uh, well, absolutely sort of crucial for anything to go ahead in this research. So thank you, Amber, for your support and encouragement and help. And this is my team. So at the moment, I don't have a collection of photos, but I mean, you can see some names here. Okay, I stop here and let's have a discussion. Any questions?
Yeah,
Who's actually?
hi,
Yeah,
Dr. Tony. I have a question. Can you hear me
Sure.
okay?
Yeah.
Hi, it's Tony Altar. No, I thought the last slide was really intriguing about the different genes for which acetazolamide improve function. Is there any evidence that there's a microRNA that normally inhibits the expression of those same genes that perhaps acetazolamide inhibits?
I don't know. I don't know. I mean, yeah, like I said, I mean, at the
Uh,
moment,
The
there's
reason
not.
I bring it up is there are, the microRNAs often, they'll hit whole classes of genes.
Yes.
And if a drug inhibits that microRNA, you disinhibit the
Mhm.
remaining allele that can still be expressed, let's say in the case of SLC6A1. Or maybe
Yeah.
some one way to look at it, there are databases that, tell us which microRNAs inhibit which genes, and that might be a way. And there's even, I think, a pharmaco microRNA database that could be looked at for acetazolamide just to see if there's any overlap with these same candidate genes.
Okay. So is there like, I mean, are there like drug effects on microRNAs?
Oh, yeah.
Okay.
Yeah.
Maybe you could send me a link.
I haven't looked at those databases in a few months, so it's not on the top of my mind. I'm just suggesting that it
Yeah.
has potential. You have
Okay.
multiple genes that this one drug has an effect on them all. Maybe there's a commonality through the microRNA.
Of course, that would be a really like exciting possibility, which I must say has not occurred to me. Although I'm just like during the last, I don't know, 5-6 years, I mean, sort of like approaching a little bit like genomics, but otherwise not.
I think it's an interesting concept because, as I recall, SLC6A1, it's a haploinsufficiency, it's a heterozygous mutation. And
Mhm.
so, right, so that means there's another allele that's still functioning. So anything that you can do to augment the function of the remaining allele, could compensate for the loss of function of the mutated allele.
That's interesting. And also, what seems to be the case that proteins are pleiotropic. I mean, which of course drives the whole community crazy because I mean, many proteins are, for instance, through the prenatal period, they have completely different functions, and they may be pathogenic. And then you see just the phenotype at much later stages. So there's more and more information on this kind of like pleiotropy before
Yeah.
birth. So yeah. So should I actually like who's the chairperson here?
Okay, is it time for the next question?
Sure.
Okay, I believe it is Michael Kavanagh.
That was a great talk, Kai.
Thanks.
Could you maybe touch a little bit more on the relative permeability of bicarbonate and chloride through GABA-A receptors and the reversal potential? And you
Yeah.
probably have done this, but if you're recording acute GABA-A currents in slice say, and you apply acetazolamide, how does that change the reversal potential of the GABA?
Can change it. This actually depends on how much you put load on the because bicarbonate is there. It's there like at steady state something like 50 millimolar. But then if you have if you activate a big conductance, of course it gets depleted, and this is when carbonic anhydrase starts to be like really crucial. But regarding the permeabilities, I think people are, I mean, there was this Bormann paper which has been cited all the time, like they never looked at physiology of this thing. So they had this, in a biionic situation, they had a permeability of 0.2. But this doesn't actually mean a lot because, I mean, one should calculate the permeability from the Goldman-Hodgkin-Cots current equation and not from a biionic like equilibrium situation, because these are totally different. And we measured this we measured this actually in crayfish. It's the channels are evolutionary. The filter is really well conserved evolutionary. So we got a permeability of 0.4. And obviously, I mean, this chloride and bicarbonate, they behave totally different within the channel. So so I mean, I would say that there's like a really big influence which is ignored. And then if you multiply, say, the internal chloride, which is 50, and you multiply it by 0.3, you get 3 millimolar, which means that you have like an equivalent of 5 millimoles of chloride just by the bicarbonate, which then means that you can't have E-GABAs which are more negative than -80. Because KCC2 is at equilibrium at 3 millimolar. So, but I mean, people measure like even -100, but that just can't be true. I mean, that's the even not possible thermodynamically for KCT. So yeah, so I wish people would like be a little bit more like flexible in their thinking and just accept this as a fact.
Okay, next question is Pasquale Striano, and then Carol, and then Bruce Bloom.
Thank you, Amber. Hello, hi. Thank you very much for this outstanding academic lecture on this topic. I really appreciate. You know, with fantastic Georgian colleagues, we are observing, collecting data on a broad spectrum of different genetic encephalopathy that are showing that acetazolamide effect can be very impactful, not only on epilepsy severity and seizure frequency, but also on other symptoms such as ataxia. So my question are two, and I think are related. The first is there a potential age-dependent responsivity to this mechanism? I guess not, but I want to check. And the second, this is the clinician perspective. Can you encourage us to anticipate in our therapeutic algorithm the use of acetazolamide, which is instead used as a last resort, or as, you know, as only after failure of several seizure medication? Thanks again.
So should I should I evaluate the possibility of just using acetazolamide as a first-line drug?
Yeah, I mean, maybe first line or second or third line, but not last resort. That's what I mean. Do you think it makes sense?
Absolutely. And what I've been actually heavily involved in like neonatal seizure research, and also like we are writing with Wolfgang Löscher and Alistair Gunn. We are just writing a review on seizure suppression in neonates, and this has of course been a real problem. So it turns out that people just stick to phenobarbital, and obviously like something like which is really benign. I mean, acetazolamide is not a really heavy drug. So my recommendation would be, in many cases, why don't you first try a very, very like light drug, like it's actually really safe. I mean, acetazolamide is a very safe drug in short-term use if you talk about like, and then try the heavier ones. Because once you have used one heavy drug, the seizures are not anymore what they used to be. Like, I mean, this is also like what annoys me in the neonatal trials. First you give phenobarbital. The seizures are phenobarbital seizures after that. They're totally different. But I mean, of course, the with these sort of encephalopathies or like NDDs, it's a different situation because you need a long-term medication. But to cut the long answer short, yeah, why not use something light first and then go to things that change, like the mild, yeah, that interfere directly with neural signaling, like action potentials. I think that's extremely dangerous.
Good morning. I guess I'll go now. Thank you for your presentation. It was really great.
Thank you.
I have a question for you or maybe for the rest of the community, and it has to do with the phenotype of the GAT1 knockout mice, because you were showing that one paper from Istvan Modi's group. Where the reason I'm asking too is because we're actually writing a paper right now trying to look at the phenotype of like comorbidities that happen in the S295L knock-in mice. And I've been doing a literature search to find all that's known about behavioral comorbidities in the GAT1 knockout. And
Yep.
I found obviously the paper you were looking at, but then I also found a whole bunch of papers, probably about 6 from different investigators in different universities in Shanghai, China. And I think they made their own knockout and they had like a different phenotype. So they actually saw increased locomotion and reduced anxiety, as opposed to Istvan seeing that the mice moved less and they had more anxiety, kind of closer to the patient phenotype we're seeing. So I was wondering if you or someone else in the community kind of knows something about, why there is that difference, and if that's been observed in those 2 knockout models. I mean, it should be the same, but I know they were created differently. So maybe there's some details that are known that I'm not aware of. Do you know anything about that?
I can give a very general answer. I mean, background. I mean, basically the whole animal is different, and this like there's so much like epigenetic and also genetic drift, but mainly epigenetic stuff. So just as an example, ApoE4 is sort of it's bad for you, but then you go to some like subsistence cultures. There's a lot of research on them. So ApoE4 is actually beneficial for them. So just to give a human example. So I mean, it's very hard to like, yeah. So very often it depends in which environment you are looking at the gene's function and outcomes and so on. But it's of course interesting to have this.
Have you considered breeding those on the collaborative cross? This is Paul Kearney. Have you considered breeding your mice on the collaborative cross to expand the genetic diversity?
Did you ask Kirill or me?
Yeah, whoever. Yeah.
We haven't even had the mice. So
Right. So one of the problems with inbred strains is, it's like studying one person, right?
yeah, sure.
Right. So you have to expand the background diversity, and the collaborative cross, that resource is at UNC and Jack's lab, or the diversity or the outbred, right? Gives you that diversity. So if you look at just what Jennifer Kearney, for example, does when they just change the background, 129, C57, you get a different phenotype in the case of Dravet. And this has been shown in several models, including the work in autism. And there's some papers in Neuron and what have you. So we've started doing this, a lot of work, but you use different, when you do deep phenotyping, say in Dravet or other backgrounds, you get a whole different phenotype, but it's more expressive of what you would see in a human in terms of heterogeneity. And you find that the primary mutation really is highly dependent on, say a GABA mutation, say in the work of Laurie Isom, who has looked at this. So it's a lot of work, but, these mouse models have taken a lot of hits because they don't really translate over to human unless you start adding the background diversity, but it's a lot of work, a lot of breeding F2s, RNA sequencing or the QTL, and then you drill down to the RNA and you get your target.
Yeah, Michael, that's a really good point.
But it hasn't been, I don't know if the communities, as the community moves away from mice and more towards cell systems, because you can get a lot of this just by getting stem cells, right? Right, but you don't have a, yeah, so it depends what your goal is.
Yeah, well, the community should move back to rats. But
well, they are, and there's some of these mice now, rats now, you can add diversity rats, but the NIH, the Systems Genetics Consortium, that was a $400 million enterprise to build that with the with Oak Ridge, so I don't think they're going to replicate that. Anyway, yeah, so just an idea.
Yeah, thanks. Yeah. No, I mean, that's absolutely important to remember what we are looking at.
Well, I'm a clinician scientist, and I admit that I just don't see those phenotypes in human. I see it in a few patients, but not across the board. So, and where we do see it is in these N of 1 studies, where these are ultra rare. We're studying Baker-Gordon syndrome also. And there you do see it. Yeah, but you have to play around with diversity and work with people that really can give you that gene network perspective from a systems perspective.
Yeah.
Look at the epistaxis, or like you said, the environment. And the environment has to be, of course, a natural environment. They can't be just toys and stuff, right?
Yeah. Just a brief book advertisement. There's a fantastic book by Nahum Ulanowski, and the title is easy to remember: Natural Neuroscience. It's really something that makes a lab behavioral person to completely rethink everything. So
I've read that book. I agree. I totally agree. When I read that, oh my God, I see it. For example, when I was at UNC Chapel Hill and moved to St. Louis, we didn't see the same phenotype. When we changed, I'm like, well, we didn't see that phenotype at UNC, and the UNC phenotype was the behavioral environment was much, I will admit, much better. Okay, much more natural, if you will. Yeah.
Yes. Yeah. I mean, those. Yeah. Yeah. Oh, yeah. We could actually talk about this a lot, but I mean, yeah, it's definitely important to keep the limitations in mind and also possible wrong conclusions.
I think I'm next. Great talk. 2 questions. If you give the drug that you're suggesting, and does it have effect? Would that have effect on other drugs that are being given at the same time? I know that there's a lot of drug interactions with acetazolamide because of how it's acting. So what would you have to be careful of in treating patients who are on poly medicines? Actually,
I don't know, because I mean, so far, I mean, this should be, I think there are really good drug interaction again databases, but I have to say that I just joined the now I'm sort of giving the but I actually joined sort of post hoc the clinical people. I was lucky to have the lucky to have this possibility, but yeah, maybe you should ask Gia or Pasquale about
We'll just have to keep track of it if we actually give this to patients. And a quick second question about a drug that does tonic inhibition like gaboxetol. What would you think that would do either alone or in addition to something like acetago? I'm seeing the word in my head. I can't say, but whatever the drug you were suggesting.
So an activator or inhibitor of tonic GABA.
It's an inhibitor of tonic.
So it would obviously, I mean, the ionic load would be smaller than the neuronal acidification would be. The neuronal acidification would be smaller, but yeah. So that's just a very fast response.
Thanks.
So there are lots of Amber, there are lots of hands.
I know, I'm not sure what to do, because there are still more than 40 people on the call. Per
Yeah,
Perhaps people interested in still discussing could just go on video and unmute and we can talk. I know well, I was going to say her last name, Pam, but we're just going with Dr. McDonald today. Maybe you can go first because I'm sure you're about to run to clinic.
Yeah, thank you. I have a bunch of meetings. No clinic today, luckily, but I just want to put in my 2 cents, two comments and a plea to the bench scientist. I'm a clinician. I have used acetazolamide for a long time for management of pseudotumor cerebri, now known as idiopathic intracranial hypertension, and I have also used acetazolamide for intractable management of absence and generalized epilepsies like juvenile myoclonic and juvenile absence epilepsy. And my 2 comments are acetazolamide. I always get very excited when there's repurposing of drugs that I'm comfortable using. So thank you for bringing attention to this. However, I would caution that acetazolamide is, although we use it, it's not necessarily a benign side effect profile. And I always get very nervous using it with patients that are nonverbal or have poor verbal skills, because in my patients that are neurotypical, I tend to reach an intolerable side effect profile pretty quickly, characterized by nausea and tingling and not feeling well. And so that's, one comment I just wanted people to keep in mind. The other one, just to touch on the neonatal seizures, acetazolamide wasn't necessarily trialed in neonatal seizures, but bumetanide was, Bumex, which also sort of has a similar mechanism of action. And really, those results were not great. There's been some concerns about hearing loss, but also just lack of efficacy, and
yeah.
just keeping that in mind.
Bumetanide is a total failure in this, because it doesn't even get access to the brain. I mean, we have, like, I tried to, in several papers, with data and with all kinds of, there's no point in using bumetanide. And also, if you want to target brain NKCC1, the vast amount, the major part is in oligodendrocytes. So this is totally alien to the people who use it for, but you wouldn't even want to bumetanide to go to the brain because it would really damage the brain. That's for sure. So
yeah, well, I think it was tried. But so just to there's a lot to do in neonatal seizures, but I would caution just from that perspective.
Okay, great. Can you maybe send me some material?
Sure. My plea more than anything is, we love to count seizures. Is the conversation I'm with my patients all the time. And I'm a epileptologist. I love to count seizures. It's easy. I can do, well, not always so easy, but with the EEGs, we can quantify, we can, run these tests. However, as everyone in the SLC6A1 community knows, that seizures are not always necessarily the major clinical outcome that everyone wants to see. And I really liked how you were showing how the electrical current improve, and that's great. But as we're doing our trials with mice, perhaps to move on to other outcomes as much as possible with behavioral, mimicking, what we can, because that would really be a really good path forward for us clinicians. Right now, I do have people coming to me who perhaps come here or they talk to other people in SLC6A1, and they want to try all these drugs that have been kind of used in a few people here and there. And of course, as a clinician, it's hard for me to want to do, one thing or another. And so that's my plea to the bench scientist. And I know people were asking about drug-drug interactions, and I just did a quick search on lexicon, but acetazolamide interacts with amantadine, which is one of the other drugs that I think has been proposed to be used in the SLC6A1 community. So thank you for looking into repurposing drugs that we are, already comfortable using. And I would love to just get more data. So really appreciate that.
Okay, great. Sounds, yeah, thanks. Very, very encouraging and very informative as well. So and also caution is needed. So yeah, I hope we can stay in touch, because
absolutely, yeah,
you have lots to, you seem to have a lot of experience. So I start to give.
Lindsay, oh, go ahead.
Oh, sorry. I just wanted to comment on the issue of bumetanide. Because I'm familiar with the studies that go into that, and it's actually been found, especially that in Boston here, with the study led by Janet Soul, that at least in those children who had the highest seizure burden, that there is actually a benefit of bumetanide, but only in the children who got the highest dose level.
Right.
Now that study had only about 60 people total. And it took about 10 years to get that many. And so
Yeah,
it just kind of warns that there's, clearly, if a form of bumetanide or something similar that could get across the blood-brain barrier a little bit better, that would be fantastic. But it does help those kids with a greater burden, seizure burden, or at least it warrants a larger study to figure that out. I'm
Right.
curious to know what is the reason you might think, Professor Kyla, that that bumetanide would be so detrimental?
Because I mean, just 5% of the brain's NKC1 is in neurons. Maybe
Oh, so something more specific
it's
to
actually
bumetanide.
it's totally. I mean, the pharmacodynamics is totally messed up. And the other thing is that bumetanide does not get access to the brain at concentrations where the therapeutic absolutely doesn't, where the therapeutic effects have been seen. So my explanation is that bumetanide actually it reduces peripheral inflammation, and that may be the sole reason. Like
So,
not,
well, I would just point out that it did help the children in the Boston study. I'd be happy to send you that to your email
I
if you.
actually have seen it, and
Right.
also
Oh,
I've
you
written
have.
about it. But I mean, they actually
Yeah.
they did a post hoc. I mean, you don't do clinical studies and change the goalposts. I mean,
So,
that's a no-no. And the meta, really, I mean, I'm actually getting a little bit like excited here because I mean, I'm so against bumetanide because of basic science reasons and because of pharmacodynamic, pharmacokinetic reasons. And of course, I mean, if you change the clinical trials aims post hoc, then the whole trial loses meaning. They
so
can cluster and recluster like they whatever they want to, but I mean, it just isn't not like
Well, I agree with you, Professor. It's I was only peripherally involved with it, just
yeah,
enrolling some patients, but it took 10 years to get 60 patients, and so that was the reason why
yeah,
they had to try to give it over to the statisticians and ask. And I gotta admit, I don't understand exactly 100% what the statisticians why they did what they did, but what they did was they looked at seizure burden because
yeah,
there were too many levels of treatment, four separate groups
yeah,
in that study that control in three different doses uh,
right.
between 60 patients. And so that was the reason why there was, they had to do some things post hoc. But, it at least, it seems to say that for those kids who have the highest seizure burden, the bumetanide did actually have a positive effect.
Yeah,
But one
right.
other thing I'd point out is that the study was designed to look for patients who failed phenobarbital treatment. And what some other studies have seemed to indicate is that if you find those patients early on, like with early EEG, and attempt to give them bumetanide or rather phenobarbital. I think what I'm thinking of specifically was one where it was looking at, if I remember correctly, it was levetiracetam versus phenobarbital, that study was designed so that as soon as you see any activity, or perhaps it was even at risk, I'll have to look it up and find out the details. But the point is, very early on, giving phenobarbital actually had at least as good an effect as levetiracetam, much better than what would have been expected, and that was probably because the patients were found so early. So I think that the main point of that Janet Soul Boston bumetanide trial isn't that bumetanide definitely works, but rather that bumetanide is worth looking into, and especially if instead of in that study where it was designed to give a dose of phenobarbital and only in those kids where phenobarbital had failed, then give the next dose of phenobarbital with bumetanide. You're in a sense, if early intervention is what's necessary, you're almost guaranteed to fail, and yet it still helped those kids with the highest seizure burden. So perhaps in a different trial, in a larger trial, where you were giving the bumetanide earlier, it would certainly be worth looking into. Especially, I mean, you raise good concerns. I mean, if it could be targeted to neurons, if you could get across the blood-brain barrier better, these would all be good things. But I think it's not something to write off at this time.
Okay,
Thank you.
right. Thanks.
Hi, sorry, do you mind if I jump in here and ask a question? Can I ask a bit of question about the bicarbonate ions? So go into a little bit of the biophysics. I know you mentioned there that we shouldn't ignore them. And I must say, since I've come into this, since I started talking about gain of function variants, I've noticed people like to give me one reason why these GABA receptors cause all these different things. And I think that's probably wrong. I think sometimes it'll be because of current verses, sometimes it's because of disinhibition. But I want to go and talk about bicarbonate for a second.
Okay.
So, how would you tease out where the bicarbonate is causing the problem and where chloride ions are causing the problem? So for instance, are there any particular residues that cause a difference in the selectivity between bicarbonate and chloride ions that we could go back and look at our patient population and see, oh, maybe we've got a mutation in that M2 region, just out of curiosity.
You mean like changing do you mean residues in the GABA
Yeah,
channel? Well, the
yeah.
GABA channel is like the ionic filter is extremely conserved. So to my knowledge, there are no mutations or no variations there. So it's something like really like
Oh no,
absolutely.
but we have patients with variants there. Yeah.
But I
So
mean, in the ionic filter, you mean?
yeah, yeah, like there's kids with those. I mean, they're very, very sick. But the question is, are there any variants there which might have cause a difference in the selectivity between bicarbonate and chloride?
My suggestion is here, because I mean, like, yeah, I have no. I mean, this is absolutely interesting. So can we continue like the? Can we
Yeah,
continue?
I mean, I was just saying that it's
Yeah.
like, yeah, I agree with what you're saying.
Yeah,
What
so
I'd
please,
like to know is how can we
please,
tease that out?
you're in Australia, so you probably are not going to continue after this Zoom meeting. But
No, no,
anyway,
I'm going straight to bed. I'm lucky to be coherent right at the moment.
yeah, but Nathan, please send me an email. Like
Yeah.
this seems to be a really nice, like a fruitful, fruitful meeting. I'm learning a lot.
Cool. Okay, I will. Kai, yeah. All right.
Yeah,
And I'll make a note of it now so I don't forget.
good. Yeah, please do. Yeah, yeah. Excellent. So Michael has something.
Yeah, hi Kai.
Hi.
Michael Rogowski here at UC Davis. One comment and a question. Um, I wonder about the pharmacokinetics of acetazolamide. It's pretty slow to act, and I think that's because it's slow to be absorbed, but also because it's slow to cross cell
Yeah.
membranes. It's not a particularly lipophilic compound, so it takes
Yeah.
time.
Right.
And I wonder if you've considered that in relation to use in the treatment of neonatal seizures, where you need to really get an effect quickly. If you had to wait an hour or two or more, that just could be practically a difficult issue.
You're right. One of the reasons is that the acetazolamide has been like already used in neonates, and it's available as a sort of IV solution. Those are really like a heavy points. The other, well, I mean, you could just take a more lipophilic derivative, but then you would need, I guess, all kinds of like permissions and yeah, and also like I think it would become a totally different game.
Yeah,
So
yeah.
those
No,
clinicians,
obviously improving on acetazolamide would make sense, but as you say, it's a major effort to do that. Yeah, let
yeah,
me just ask my question, which is really a very simple kind of question, and that is you made the really great point about the need to correlate the developmental stage of the rodent versus the human. And I'm wondering, just in a standard human at birth, what is the dynamics of chloride? Do you really believe that at birth, in humans, GABA is still depolarizing, or
No, no, no. I mean, it seems that it's we are actually the human is born at a very strange like time window. I mean, like for instance, the KC2 expression is like it's starting to plateau, so it's not really there. But
See, that would make total sense to me because it's already been mentioned many times that phenobarbital is the drug of choice to treat neonatal seizures.
it
And
definitely it's not really
and
being
and you're
explored.
you're saying that you'd like to have an improvement, and I think that's reasonable. But as it was also, I think Kieran mentioned, when it was tested in relation to levetiracetam, it failed the levetiracetam. So phenobarbital actually, this is kind of an interesting historical anecdote. Phenobarbital was introduced into epilepsy treatment in 1917 or something like that.
Yep.
And it was never FDA approved because it was grandfathered
Mm.
when the FDA was created. It had already been in widespread clinical use, so they had these grandfather provisions. And I think it's one of the last grandfather drugs to actually be now approved. It was just approved a couple of years ago.
Yes.
And its approval was based on that study that Kieran mentioned in neonatal seizures, where it clearly showed an effect. So yeah, we need to improve on phenobarbital, but at the moment it's the only drug we have that we know actually
Yeah.
is effective, and that's why it is the drug of choice at the moment.
Yeah.
But
Yeah. I would like
uh,
to make a slight addition here. I think seizure suppression is useful in neonates only if you can show that the outcome, the long-term outcome is better. And all the studies I know from like with outcome is that actually the outcome is worse in animal models. And then there's also some human data showing that in fact the phenobarbital is not really good for the immature brain, like people know from anesthetics that they really screw the brain. So you can get rid of seizures, but I mean you can cause more damage with phenobarbital than with the neonatal seizures. In fact, nobody has shown that neonatal seizures per se are damaging, but there's there's quite a lot of data that phenobarbital is. So I think like it's, it's such a
I'm
strange
printing
field.
just one paper, so probably not all is mine.
Sorry?
And listening to a webinar at the same time, so that's easy, right?
Yeah.
Mhm.
Okay, are we?
yeah, I just wanted to make one comment. I mean,
Yeah.
I think Kai, you raised some really interesting issues around phenobarbital. It's definitely a drug that we've put to the side in the treatment of epilepsy generally because it is so fraught with side effects, and perhaps there are even worse problems in the neonatal period that we're not aware of. But I think it's an interesting proof of concept because, of course, phenobarbital works mainly by enhancing GABA-A receptor currents, and probably also directly activates currents that may enhance the tonic current. There hasn't been a lot of research done to explore that. But um, if GABA was depolarizing at birth, and you gave a drug that enhanced GABA
Mm. Mm.
uh, A receptor activity, it shouldn't be anticonvulsant, but it is. So I think that confirms the idea that at least at birth, it's gotten to the depolarizing activity has waned, and it's mainly hyperpolarizing, or it clamps
Yeah.
the
Right.
reversal
So
the
yeah.
reversal potential for chloride.
Right. And if you go like slightly one step further, it's not so much about the reversal potentials, it's actually about the chloride loading, which I mean, like the sort of the voltage response per se is what's actually the problem is that during phenobarbital, you get accelerated loading of the neurons with chloride in an activity-dependent manner. And so it's a very sort of a very, very dynamic situation where things can go really wrong. So so that phenobarbital really cements the seizures, despite the fact that KCC2 is sort of relatively like highly activated, but it can't do anything anymore when the cell is full of chloride. So it's always a load. It's a leak load, or actually leak pump or load pump relationship, which governs the effect of such drugs. But these are never discussed in literature. It's only about this hyperpolarization or polarity. But it's basically it's a classical pump leak relationship, which the Danish researchers showed like 100 years ago, but which has been happily ignored in the chloride field.
Actually, can I jump? Oh,
yeah.
So sorry, I just want to jump in there real quick because that's exactly what was being discussed right now is exactly what we were talking about with the as we understand bumethonide's function is that when you use phenobarbital and you know like enough time has passed, it's because of the chloride loading is how we understand that. If you can stop that chloride loading, intracellular chloride loading with something like bumetanide early on, then phenobarbital can have its desired effect. It can then be efficacious for patients. That's exactly how we understand that relationship. So I just wanted to point that out. Yeah.
Yeah, but one point here: the loading doesn't come via NKC1. The loading comes via channels. So the main, I mean, yeah. So I mean, yeah, these like triangular cells which have KCC2 and NKC1 and then channel. Forget about them. I mean, they're just rubbish. I mean, talking to a quite large audience that maybe uses these schemes, but these schemes are really misleading. I mean, they don't get the dynamics right. So most of chloride loading in active neural networks comes via channels. And unfortunately, there was well, in 1999, there was a preview, I guess, of our Nature paper. And this preview had this first, like, whoever wrote it had this, like, a triangle or whatever. It was with water filled half. It's just nonsense. I mean, it's not transport physiology. It's basically, no one working in the pH field would do anything like that. So the pH field is much more informed about ion regulation than the chloride field. And it's very hard to bring the pH field's expertise into the chloride field, which is one of my missions.
Can I just ask you, I'm sorry, I didn't mean to like skip the line, but so the idea though is that we're not talking about steady state, we're talking about like an active seizure, and that then if there's an active seizure going on and chloride would be allowed, you get chloride loading then as a result of GABA activation inside the cell.
Yeah.
Sorry?
Yeah.
Yeah.
Sure. Yeah, just to make, I mean, okay. So we had a paper with Wolfgang Löscher in an NKCC1 knockout, total knockout, and looked at KNet seizures. They are even worse in the NKC1 knockout, total knockout. And now we have in the lab, we have a forebrain principal neuron knockout, NKC1 knockout, no NKCC1 in the pyramidal neurons. So I have some preliminary data, but I'm not going to publish them now. Then we also have the next one is we have a tamoxifen inducible like oligodendrocyte specific knockout. And I think this is an interesting comparison because I mean the latter has a lot of NKC1 and in the oligodendrocytes, but neurons really don't. So and yeah, so anyway, so
Yeah, I guess I would just point out that if you have a pathophysiological situation where you have chloride loaded inside the cell, it could then be that whatever contribution of NKCC1 that you would be having in that neuron could then, by blocking it, in that pathophysiological state, this could be very therapeutic, where you wouldn't have seen a big result in the physiological state. That's all I'm saying, and because clinical evidence.
Yeah, that's a possibility that's been like, yeah.
Anyway, I'm sorry. I just because it
No
sounds exactly
problem.
the same. Yeah.
Fun to talk.
Thanks, Guy, for your talk. I really learned a lot about acetazolamide and bicarbonate. So my question is, what is the blood-brain barrier penetrance of azathioprine? And do you know there any off-target or like systemic peripheral effect of this drug, and that might contribute to anti-seizure effect?
There are a lot of peripheral effects, obviously on the kidney and so forth. So it does have, as Pam pointed out, lots of side effects. It does cross the blood-brain barrier, actually, but as I said, Henry, it's quite slow. And I think that could be an impediment to its practical use because it takes, an hour or two for it to act. So if you gave it intravenously to a neonate, you'd have to wait around for a while for it to work.
Yeah. So of course there's this initial sort of respiratory acidosis which is produced, which is also like suppressive seizures. So in our like rodent model of asphyxia, it's actually the respiratory acidosis which blocks the experimental seizures. But of course, these seizures are much lighter than those seen in babies, so human neonates. So I guess the sort of the translatable part of our model is mainly based like on modulation of seizures rather than like a strong suppression of seizures.
Another problem that we haven't talked about with regard to acetazolamide, I wonder what you think about this, Kai, is the fact that there's a tachyphylaxis to the effect. It only works for a period of time, and then the kind of acid-base mechanisms compensate, and it stops working. And that's actually one of the reasons why it's not used clinically in the treatment of epilepsy. It is occasionally used in the treatment of catamenial epilepsy, which is seizure exacerbations at the time of menstruation. And there you don't have to worry about tachyphylaxis because you're just giving it during the vulnerable period. But if you tried to give it chronically, it stops working.
Yeah, right. But Michael, so I mean, if you say that there's tolerance developing to acetazolamide, so would you take like 1 year as a like criterion of tolerance, or many years, or a few weeks? Where does the tolerance criterion, where does it actually come into play?
I
Because
have to look that up again, but my recollection is that it's fairly quickly. As I say, over a period of a month, you could give it just during the few days before menstruation, let's say, and during the menstrual period, and that's the way it's used, and then it will reset. You could give it again the next month.
Right, but in this like in a manuscript which is being now written up, so I guess the fastest tolerance develops in 1 year, and there are some people who have been 11 years on acetazolamide. So I think it depends a little bit on what the problem is, like the CNS problem.
Yeah, that's true. I do use it in some patients to treat idiopathic intracranial hypertension, pseudotumor, and they can get a benefit over a period of extended period of time. So it doesn't always show tachyphylaxis. I agree.
Yeah, and the tachyphylaxis for phenobarbital can be actually after a few seizures in the neonate. I mean, I can send you some interesting data which we are about to.
Well, that's true for benzodiazepines in spades. They
No, but I
really
mean,
show.
but with phenobarbital in
phenobarbital also interesting.
Human neonates, yeah, that was quite surprising. Like, I mean, yeah, we got the data from Geraldine Boylan, so I mean, it's quite sort of impressive. So.
And I have another question. So in relevant to SLC6A1, one of the I think the major or one of the major circuits for this seizure is the thalamocortical connection. So I'm wondering if the drug might be more accessible to some brain region that might more engaging that therapeutic target. Do you know if anything about that brain region specificity or even cell type specificity, which might be Michael is asking question next.
Henry, the acetazolamide actually blocks the seizures and the ataxia in these patients. And it occurs, strangely enough, it occurs simultaneously. So there seems to be like a thalamic involvement. There seems to be a cerebellar involvement, and the cerebellar involvement was obvious from the classical studies by Moody and Lester. So yeah, fits sort of quite nicely. So
that's right.
A quick related question. Are there differential effects of the drug on GABAergic and glutamatergic neurons?
I don't know. I don't know. I mean, yeah. So yeah, inhibiting inhibitory neurons wouldn't be a good idea, right? But, yeah.
Well, you have the rat model to test all these hypotheses. I think that would be good.
Hi, everyone. Is that the last question? I think so. Amber had to leave. I'm Lindsay from SLC 601 Connect UK. Hello. And if there's no further questions, I can thank you all for coming and for participating and for, you know, the plethora of questions that came around that amazing talk. Thank you so much, Kai. And
For the opportunity, really enjoyed this.
We enjoyed it too. And yeah, Amber will send out the recording generally on the next email letter and the invitation to next month's meeting.
Yes,
thanks
everyone for your patience and interesting comments and kind remarks. See you later.
Thanks, everybody.
Thanks for the great talk.
Thank you.